Hello students, welcome to SK Biology. Today's video is about genetic code. Genetic code is the set of rules by which information encoded in nucleotide sequence, that is DNA and RNA, is translated into amino acid sequence of proteins. This information is read as set of 3 nucleotides called as codons. As we know, DNA contains 4 nucleotide bases ATGC and RNA contains AUGC. Codon in genetic code contains 3 bases which code for one amino acid, and these different codons leads to synthesis of 20 different amino acids which are responsible for synthesis of protein. Now the question is how 4 nucleotides code for 20 amino acid. To find answer to this question, scientists proposed 3 assumptions. First assumption is one nucleotide is coding for one amino acid. But in this way, we will only get 4 different amino acids, not 20. So this assumption is discarded. Second assumption is code is doublet, that is having 2 nucleotides. This also giving 16 amino acids, not 20. So third assumption scientist given was codon is triplet. This assumption can give 64 different codons, and now it has been proven that 61 codons are for amino acids and 3 codons are for stop codon. Once the idea of codon cleared, scientists cracked genetic code by doing different experiment. The first experiment was done by scientists Nirenberg and Mathai. They did in vitro protein synthesis by single nucleotide inclusion, that is one nucleotide at a time. In experimental setup, they have taken 4 test tubes and added all the components needed for RNA and protein synthesis, like ribosome, tRNA, mRNA, E. coli extract, etc. In each tube, single nucleotide is added. When this reaction mixture undergo transcription, it is found that in adenine containing tube, homopolymer of adenine is synthesized. In the same way, homopolymer of cytosine and homopolymer of uracil is also present in respective tube. But in G, they found quadraduplex structure, not homopolymer. When these reaction mixtures undergo translation, poly A tube gives rise to polylysine peptide, poly C produces polyproline peptide, and poly U produces polyphenylalanine peptide. Due to quadruplex structure, G is not able to produce any amino acid. In this way, Nirenberg and Mathai gave first experimental evidence which linked specific codon to specific amino acid and helped in deciphering genetic code. Next experiment was done by scientists with heteropolymer, in which they have taken fixed proportion of nucleotide to synthesize heteropolymer. As here in example, 1 part of uracil and 5 part of cytosine will result into synthesis of U once and C 5 times after transcription. Translation of these 6 nucleotide containing heteropolymer leads to synthesis of serine and proline nucleotide, which suggests that the first codon UCC is coding for serine and triple C is codon for proline. In this way, code for all 20 amino acids has been identified. Next breakthrough in the study of genetic code was given by Nobel Prize winner Indian American biochemist Hargovind Khurana. Hargovind Khurana developed precise technique for the synthesis of heteropolymer with complete accuracy. He used repeating nucleotide to produce polynucleotide. Then polynucleotide was translated to identify amino acid. First, he used dinucleotide UC. The resulting polynucleotide is UC-UC repeating unit. With this heteropolymer, only 2 codons can be produced, CUC and UCU. These codon gives rise to serine and leucine respectively. Then he used trinucleotide GUU. The polynucleotide produced is GUU repeats. This heteropolymer resulted into formation of 3 codons GUU, UGU and UUG, and these respective amino acids for codons are valine, cysteine and leucine. Such synthetic polymers were later used to decipher complete genetic code. Hargobind Khurana got Nobel Prize for his important contribution in the molecular biology.